Okay, I think we'll go ahead and start. So welcome everybody. Welcome to the Department of Energy Isotope Program Actinium-225 User Group Meeting. So we like to have these annually so we can kind of give you a kind of an update of where we are, but also so we can have some people who use the product kind of give you their experiences and then get some feedback from you. So the way that this is going to work is we have 4 speakers, and the 4 speakers are going to give their presentations. And then at the end of that, we will open up for questions and answers. So if you look at the bottom of the screen, there's actually a Q&A button. So as we go along, you can put your questions in there as we go through, and then we'll pick those up after we finish the presentations. So those of you who don't know me, I'm Cathy Cutler, and I'm the director of the Isotope Program at Brookhaven National Lab. I'd like to introduce the other speakers. I don't see Gary, but Kevin Roland is here from Fusion Pharma. Kevin, if you want to raise your hand and say hello. And then we have Ken Song from Razi Bio.
Hi there. Nice to meet everyone.
Thank you, Ken. And then we have Monideepa Roy from Actinium Pharma, and she seems to be sharing. But I don't know if you want to represent who's with you.
Yeah, I have our VP of Radiation Sciences, Dr. Patrick Broden, with me.
Okay, nice to meet you. And then we also have Gary Kohanbash.
Hi, everyone. I'm here.
Thank you. From the Department of Neurological Surgery at the Children's Hospital of Pittsburgh. And so Gary will be speaking first, followed by Kevin Roland, then Ken Sung, and then Monideepa Roy. And after Monideepa, we will start taking questions. So I will start with my presentation. So hopefully you can see this okay. So what I'll be doing is just kind of giving a little brief perspective on Actinium-225, then talking about the high energy accelerator production, where we currently stand with the status of our drug master files and our ongoing interactions with the FDA. A little bit of an update of where we are on our alternate production paths. I'm looking at Actinium-225, and then after that we will have the roundtable presentations where the others will present their talks, and at the end we'll then have an open Q&A. So I decided to start this. I was kind of curious about the market for Actinium-225, so I actually went out and looked at a variety of different market reports. And numbers I got ranged somewhere between 21% and 37% over the next 5 to 6 years. And the driving interest behind this is really the potential benefits that have come forward for the clinic based on the targeted alpha therapy, particularly the fact that it has been shown to work in patients who have no other options. Additionally is the fact that we have an increasing aging population, and so increasing numbers of patients who have cardiac and cancer ailments. What I further learned is that currently North America dominates the alpha emitter market, but growth is expected to grow fastest actually in the Asian Pacific region. So just showing that the interest in this is actually really growing. So there's been a fair amount of preliminary clinical experience with Actinium-225 and Bismuth-213, and this was really highlighted in a review article that was put together by Morgenstein et al. And in this article, what you can glean from it is that there's a number of different cancer types that Actinium-225 and its daughter Bismuth-213 have been evaluated in. Additionally, the radioconjugates that have been looked at, you can see that actinium and bismuth have been looked at conjugated to antibodies as well as peptides and small molecules, showing their versatility in the applications. And you can further see that there's been a number of patients that have been evaluated with these radiometals, and in prostate cancer alone, it was well over 400 patients. I think interest really picked up based on the very positive results that were demonstrated using the Actinium-225 DOTA PSMA 617, demonstrated patients who were non-responsive to other treatments, but then had a very high response to the Actinium-225 conjugate. And this has really led to the interest in developing target alpha therapy for a variety of different applications in both oncology as well as in infectious diseases. I decided to go out and look at where we stand currently with clinical trials. So I went out and looked at clinicaltrials.gov and Trial Bulletin, and then they were pretty much in agreement, and there was about 23 clinical trials with 5 currently closed. So the most prevalent clinical trials are ongoing in prostate. These numbers in parentheses actually are the number of clinical trials that are ongoing in those disease states. So prostate cancer has about 10 clinical trials looking at like PSMA, J591, and then do a variety of different combinations and a different actinium antibody. That was followed by studies in acute myeloid leukemia, where they have 8 clinical trials looking at actinium labeled to antibodies and then combination therapies. And there's a trial looking at the use for colorectal cancer, multiple myeloma, 2 going on looking at solid tumors, Actinium-225 looking at neuroendocrine tumors, and then finally metastatic uveal melanoma. So this just kind of highlights that the number of trials are increasing and the interest in actinium is ongoing. So Actinium-225 has traditionally been obtained from a thorium-229 generator. Thorium-229 was really derived, really what was waste that was generated in the 1940s and 50s. And that material has been harvested and then utilized to develop generators that now supply somewhere between 1,200 up to a maximum of about 1,700 millicuries per year. The amount that's used for the patients for actinium is about 2 to 5 microcuries per patient, and for bismuth it's about 1 millicurie per patient. And looking at the development of these agents and the ongoing research, what was projected is that the demand, assuming that multiple approved drugs and robust clinical R&D programs were developed, that we would be in a situation where hundreds of curies of Actinium-225 were going to be needed for a year to basically support these efforts. Now, Oak Ridge has really been one of the leaders in Actinium-225. They developed the thorium-229 generator back in 1997 from material that they harvested. And they've gone on to supply about 10 curies of Actinium-225 shipped in thousands of packages. And they supply approximately a curie of actinium on a yearly basis. Now, if you look at the graph below, what you can see is that the blue is the projected available activity of actinium that can be provided. And in green is the material that has been shipped out. And what you can see is that they've pretty much provided most of the material that they've had. You can also see that with time, they've been able to increase the amount of material that they could from these generators to supply it to external users. In red is actually the number of shipments of Actinium-225 that have gone out. And so what they started noticing is about in 2010, that interest in actinium was growing, and based on the demand, that before long they were going to get to a point that the demand was going to outstrip actually what they could do with the generator. So it was really at this point that the Department of Energy started looking at alternative methods for supplying Actinium-225. And at this time, the group at Los Alamos started looking at cross-sections for the high energy accelerator route of irradiating thorium-232 with high energy protons using the spallation reaction to produce Actinium-225. And what they showed is that utilizing a single target with a 10-day irradiation, they were able to make Curie quantities. So you can see here that from Los Alamos, which has an incident energy of 100 MeV, operating at 250 microamps, they can produce 1.3 Curies. Brookhaven, 200 MeV, 165 microamps, can produce up to 2.2 Curies. Now, they're knowing that the demand for this isotope is in the hundreds of Curies. There are plans for future facility and targetry investments at both IPF and BLIP. And LANL plans to work towards 450 micrograms, would allow them to supply 2.3 Curies per target, and BNL up to 300 micrograms, which would get you to 4.0 Curies per target. And BNL can actually irradiate more than one target. We've actually irradiated up to 2 targets currently, and we foresee that we could go up to 5 for this. So there have been other groups who have looked at the cross sections, and here you can see in blue really the cross sections for Actinium-225. But also here is reported the cross-section actually for Actinium-227, which is co-produced during this irradiation. And you can see it follows closely the actinium production, but at the higher energies, it actually goes lower. So we understand this, and we've been following this to ensure that we have a consistent low number of the Actinium-227. So based on the fact that we have these high energy proton facilities and the fact that utilizing them, we could make Actinium-225, the TriLab effort was formed back in about 2014. So Los Alamos and Brookhaven have worked together synergistically for many years, providing isotopes, making sure that by combining our schedules, we're able to supply them year round, and and recently actually served as backup to each other. Oak Ridge has the experience with Actinium-225 in doing the chemistry. So by forming this group, we were looking at developing the feasibility of the targetry, doing the separations, and then providing the Actinium-225 material. So here you see a plot of the ratio of the Actinium-227 versus the Actinium-225. And you can see how it kind of goes up and then drops off as you go up in energies. And the challenge with the 227 is the half-life, which is about 21.8 years. So as we've brought these products up, we really understand the impact of the energy and the long radiations on this time, and have basically set in limits to allow us to control the amount of the Actinium-227 that is produced. And we understand this ratio precisely and monitor the irradiations to keep the product consistently. The Actinium-227 co-product creates a unique set of challenges due to its long half-life. And part of this is due to facility license from the NRC, but also the challenges in getting rid of the waste. These challenges are not unique for actinium. Other isotope products have been developed and come up against this and have been dealt with in the long run. So when the isotope program brought this up, one of the interests was how was the Actinium-225 from the accelerator material going to compare to the Actinium-225 from the generator material. So they sent out material, and some direct labeling studies were done both externally and internally. And what they demonstrated is that the direct labeling efficiencies were comparable for this product derived from 2 different sources. Additionally, they used the material to look at the daughter bismuth-213 using the generator and looking at the performance, and it was seen that the accelerator material actually performed the same as that material that was derived from the thorium-229 generator. They further set up an FOA in which they funded studies for people to look at the impact of the Actinium-227 content on dosimetry. There was 3 different groups that were funded to look at this. And what it demonstrated is that the impact was very small. Despite that, there's still challenges that remain based on the logistical considerations. One of those is that due to the half-life, it's required from the NRC that you have a decommissioning fund and plan in place. There are discussions going on with the NRC about this to try to address this similarly as the same way that was done for the germanium-68, gallium-68 generator. Patient waste should not really be an issue for this, as it's, at least in the US, is not for most isotopes that are used. Now, understanding that people were interested in using this to go into clinical trials, drug master files were filed. And so a drug master file was filed for the accelerator material in December 2019. And then we heard back from the FDA that was accepted in January 2020. The DMF for the accelerator material was submitted in December 2020. We continue to have interactions with the Food and Drug Administration as we work to move along and scale up the product and make sure that quality is built into that process. And we are committed to making these products available to the customers, and we use avenues such as this to really try to engage and talk to them, to update them where they are, and to address any questions that they have. So over the past year, we've been working to increase the availability of Actinium-225. So we've actually gone to monthly production, routinely achieving up to 50 millicuries per batch at the end of processing, which you can see in the graph below. One of our challenges is that production is limited by processing capability and shipping transit times. We're looking to address those. At Brookhaven, we're bringing on new hot cells this year to add in a new production line at Brookhaven to process Actinium-225 and send it out. And they're looking at bringing up facilities at Los Alamos and putting them next to the IPF that will allow them to process, thereby increasing our processing capability. In fiscal year 2022 alone, we've already shipped out 307 millicuries to multiple end users. And forward looking, we are looking at additional processing capability. We're looking at bringing up another hot cell facility at Brookhaven called the CARP, and they're bringing up a new hot cell capability at Oak Ridge National Laboratory. Now, based on the demands and need, the isotope program is looking at alternate routes of production. One of those is argon, has an electron LINAC, and they've been looking at the feasibility of irradiating radium-226 with a gamma-N reaction to produce the radium-225, allowing them to use it as a generator to provide the Actinium-225. At Brookhaven, we're looking at bringing up the low-energy cyclotron route to look at the production of the P2N reaction on radium-226 to produce Actinium-225. And at Oak Ridge, they've been looking at the neutron production route, where they've done feasibility studies and irradiations on radium-226 through the 3N gamma route, deriving at the end the thorium-229, which is similar to the generator material they have currently. In addition to the Actinium-225, it's known that what is often needed is a diagnostic for imaging these alpha emitters. And there's really not been necessarily a great one for Actinium-225 or thorium-227. So Los Alamos, working with Rebecca Albergill, actually developed cerium-134. The nice thing about cerium is it has an oxidation state of 3, which allows you to mimic the in vivo behavior of Actinium-225 and determine dosimetry. And then in the oxidation state of 4, it can be used for imaging the potential biodistribution of thorium-227 and evaluating the dosimetry for that. There are ongoing evaluation batches, and the DOE actually has formed a cerium-134 user group, and that user group is meeting on September 19th. So in summary, the TriLab effort is routinely producing Actinium-225 and product is available for end users, and multiple shipments have been completed. To date, externally, we've distributed over 588 millicuries of accelerated produced Actinium-225 to evaluators. This year alone, we've already shipped out 307 millicuries. We are working with companies and hospitals in preparation to support clinical trials, and we are responding to requests for letters of authorization. The actinium content is clinically insignificant from a dosimetry toxicity perspective, but challenges do remain with the perception and regulatory compliance, and we are working on a path to really address this. Understanding that the demand is further increasing, as you've seen, we've been increasing production. We're further investing in our production facilities to increase the amounts we can produce, and investing in bringing up processing capability to ensure we have redundancy, ensure that we have a reliable product. So we are continuing our efforts to scale up availability of this important isotope. So if you have questions reversing availability or isotopes, please go to isotopes.gov. And I thank you for listening. Based on that, we'll go to our next talk, which will be from Gary Kohanbash. And just a reminder, please put your questions and answers in the Q&A button at the bottom. So Gary.
Okay, well, thank you, Cathy, and it's a real pleasure to be here today. I'm an assistant professor at the University of Pittsburgh, and my background is actually in brain tumor immunology, immunotherapy. And so I'm going to tell you how we recently got engaged in using Actinium-225. And so the title of my talk today is Glioma-Associated Immune Cells, Potential Targets for Novel Theranostic Molecules. And so just a little background on brain tumors. There are about 700,000 Americans living with primary central nervous systems. Gliomas are the most common, and glioblastoma is the deadliest and most common of the malignant brain tumors, with median survival about 1 year or a little more. And also in kids, brain tumors are the leading cause of cancer-related death. And so there's been a lot of research on improving outcomes for GBM patients, but really it hasn't gone far enough. And clearly, better treatment for brain cancer is a pressing need that we have. And so why we're interested in immunotherapy, without going too much detail, but immunotherapy has been shown to have this tail end of the curve, with patients actually surviving long-term, not necessarily yet for brain tumors, and it's select patients. But the idea, hopefully, is that we can combine with radiopharmaceutical therapy and other therapies and further see patients surviving and patients cured. And so there are many new immunotherapies being developed, but one of the major challenges with brain tumor immunotherapy are tumor-associated myeloid cells. And these tumor-associated myeloid cells include tumor-associated macrophages, myeloid-derived suppressor cells, microglia, and account for up to 30% of a tumor cellular mass in the brain. Multiple trials have occurred and are ongoing targeting these cells in gliomas, but so far most of the therapies have been unsuccessful at actually reducing these cells. And so the main point is we keep developing immunotherapies for brain tumor patients, but we still must overcome this very clear issue of this tumor-associated myeloid cells. And so I met Dr. Barry Edwards at the University of Pittsburgh, and this is how we got interested. We said, hey, for all these therapies, can we actually image these cells in gliomas, in preclinical models? And so we published this a few years back, that we used the zirconium-89 DFO on an anti-CD11B antibody, and we injected this into tumor-bearing mice. And what you can see is we observed very strong uptake of the tracer within the tumor, and this was reversed by using a blocking antibody, showing that it's specific for the CD11B cells. And in normal brain, we also see no uptake. And all this confirmed also by biodistribution analysis. And so this led us to ask, hey, what about a radiopharmaceutical therapy approach to deplete these cells? And so we say, hey, can we deplete these cells and also enhance other immunotherapies such as checkpoint immunotherapy? At the same time, we were using zirconium-89, and DOTA doesn't really chelate zirconium-89 well. So we tested a new chelator, Lumi-804, that could label both lutetium-177 and zirconium-89 at room temperature. Here, just to show the structure of Lumi-804, and we repeated the imaging studies that we did with the DFO zirconium-89, and once again, we get a very strong uptake within the tumor. And so then we started with saying, hey, can we see prolonged survival when we combine radiopharmaceutical therapy, TRT here, with ICI? And indeed, the mice with GL261 syngeneic gliomas, when receiving both the TRT and ICI, had the most prolonged survival compared with other groups. And here you can see actually just some of the tumors from these mice, that the control tumors grow really out of control. But with TRT or TRT with checkpoint, we really see a smaller circumscribed tumor mass in these mice. We performed CBC analysis, and fortunately, these doses did not observe any sign of like a bone marrow toxicity in these mice. And then we looked at actually what was happening in the spleen as a marker of periphery and the tumor, and to see, can we deplete, are we actually depleting our target cells? And so the MDSC and TAMS are two population of these tumor-associated myeloid cells that we're targeting. In the spleen, we see great reduction of these two populations. But in the tumor, although we see some reduction, it's not as significant. And we realize this is an area for improvement with the lutetium-177. Notably, when you look at the granulocytes that don't have CD11b, but could be in the same region, these cells mostly are unchanged quantity, suggesting really we have a specific targeting of the population we're interested in. So then, given that our lutetium-177 wasn't really having strong depletion of this target cells within the periphery, in collaboration with Dr. Nedra, we said, hey, maybe we need something stronger, such as an alpha emitter. And so just here, probably people on here are familiar, the difference of alpha and beta emitters, but the major difference for us was the higher energy. And so we came up with this idea that there's a balance between TAMC depletion and what's known about immunosensitization from radiotherapy. And so we believe that, and we're currently testing if alpha particles, specifically Actinium-225, can have a greater ability to reduce tumor-associated myeloid cells. At the same time, we know that the beta particle with a larger range may create additional immunosensitization in T-cell recruitment, antigen presentation. And so ultimately, there may actually be benefits for using alpha particles, sometimes beta particles, or the combination to achieve different goals. So to move on to the Actinium-225 DOTA anti-CD11b data, here was our conjugation strategy. We achieved high amount of radiolabeling. And one of the things we wanted to do was see if we can use cold antibody co-injection to increase the amount of antibody, radiolabeled antibody that gets to the tumor. So here you can see alpha camera images. In this data, we also have the corresponding BioD, not shown, but with 25, 50, and 100 micrograms of cold antibody given at the same time as our tracer, we significantly increase the amount of tracer and activity within the tumor. And interesting, and fortunately, we also, when we look at the liver, you can start seeing less activity in the liver with the increasing concentration or the increasing amount of cold antibody. And similar to the amount of uptake in the tumor, when we look at survival of these mice, the 100 microgram treatment group these mice seem to live longer than either the 25 or 50 microgram groups. So just where we're going next with this, we're trying to look at if we can better target the immunosuppressive TAMs. Some of these tumor-associated myeloid cells actually may not be as immunosuppressive as others, the M1, M2 spectrum. So we're looking at markers that have higher expression on the tumor-associated cells compared with the cells in the periphery, and important that they're restricted to these cell types. Another consideration is that macrophages, especially the M2, have been shown to be some of the most radio-resistant cells. And so we're currently looking at whether we can use radiosensitizing agent in combination with alpha therapy targeting these TAMs. And then we really have a host of more questions than answers at this point, but really looking at high dose versus low dose, differences in the alpha and beta, as I discussed, the timing, the dosing, but ultimately we still don't know, but can we deplete these cells in patients to improve immunotherapy responses? And that's why we're really fortunate to be able to have access to Actinium-225 for research purposes. Just as a brief summary, these TAMCs in brain tumors promote tumor growth and really are a major barrier to effective immunotherapy. We've shown that they can be visualized in preclinical gliomas, and that these cells can be targeted using radiopharmaceutical therapy. So, a lot of people to thank for our work, the lab, and Barry Edwards and Jesse Negro, really key to, keeping this going. And that's it. Thank you.
Thank you, Dr. Kohanbash. And we'll move on to our next speaker. Just a reminder to make sure you put your questions in the Q&A and we'll address them at the end. So our next speaker is Kevin Roland from Fusion Pharma.
Thank you, Cathy. And I would like to start by thanking the organizer for inviting Fusion today, and also for their continuous support in supplying us with good quality actinium for our development work. So I will speak a little bit about our latest initiatives in order to overcome all the challenges that therapies using Actinium-225. And I will provide you with a quick overview and status updates on our latest clinical programs. So when building Fusion, the goal was really to create a company that can overcome that can actually better utilize the power of alpha radiation. And if we look at the radiopharmaceutical therapy landscape today, most of the development effort have been traditionally concentrated around beta emitters and for limited numbers of targets, and that for decades. And companies have generally emphasized the use of one class of isotope and delivery vehicle. So the Fusion's vision is different. We wanted to create a leading company that can develop targeted alpha therapies in area of high unmet need and pursue large market opportunity. And to do so, Fusion has really developed a unique targeted alpha therapy platform that enables the rapid creation of targeted alpha therapy with multiple class of molecules, antibodies, small molecules, and peptides. But we've also gained a good expertise and control over our supply chain, which support us in executing the company's vision. So I said, the issues we have can help us to overcome challenges associated with the use of Actinium-225, and I think this will agree with me that challenges are numerous in this field. The access of reliable source of Actinium-225 remains a challenge today and will be for the foreseeable future. Availability and the generation of IND data and actually performing IND studies is also a challenge when you work with and also the access to key resources and expertise in CMC, in non-clinical and clinical supply can be a challenge. And last but not least, positioning a new alpha therapy in the line of conventional therapy can also be a great challenge. So in order to overcome this problem, the company has put a strategy forward to make sure we can position ourselves as a leading company. We've executed on different supply agreement and investment that we've done into the production of external capacity in key development area to speed up the development and the clinical development of our lead compound. We've also invested in developing our internal resources in order to increase our manufacturing capacity. And we've also partners in the field, and we are also looking at the combination studies in order to combine conventional therapies with targeted alpha therapies. So our CMC initiatives emphasize the fusion competitive advantages. So far, we've successfully opened 3 INDs with the US FDA for multiple classes of molecule. We've deployed a unique network of CDMOs in order to support the routine production of clinical product in order to support our geography. Fusion has invested in infrastructures. As you know, we are right now building a 30,000 square feet GMP facility in Canada in order to accelerate and to fast track the completion of IND enabling studies, but in also to support the early clinical pipeline of Fusion. Working with Actinium, and it requires a lot of rigor on CMC and non-clinical. And we have developed also an expertise that help us to use actually various sources of Actinium-225. We're speaking mostly here about thorium-based and accelerator-based actinium. So we are completing the evaluation and the impact of the impurity profile of new source of actinium on product dosimetry and toxicity. And we are also extensively working on the characterization of our drug products using alternative sources of Actinium-225. So the Fusion's pipeline is a good example of our ability to do the best use of alpha radiation. I'm gonna present you today a quick updates on the 3D program that are right now moving into phase 1. We also recently announced our first collaborative program with AstraZeneca, and we've released our first target, and that is now moving into IND enabling studies. I will come back on that as well. So in terms of program updates, our lead program with PI 1434 is a product targeting insulin-like growth factor 1 receptor, IGFR1R, and it's a very attractive target for developing an alpha therapy. As you can see on the right, this is a target that is overexpressed on the surface of pretty much all solid tumor cancer cell. It's also show a very low expression of the surface of normal tissue, as you can see here in the middle. And it rapidly internalize receptors in order to concentrate the alpha particles inside the tumor cell. During the same user meeting last year, we had briefly presented the results of our phase 1 single dose study for this compound. With those successfully dosed 12 patients have seen a great uptake in multiple tumor types. We studied 3 dose cohort up to 40 kilobecquerel per kilogram, and we've completed that trial with a very good safety profile. Since then, we've moved to phase 1 multi-dose trial, and we are there studying 2 regimen. One is hot only or radio-labeled only regimen, studying a dose from 40 to 100 kilobecquerel per kilogram. But the one I wanted to highlight here is our second regimen, which is studying the pre-administration of cold antibody prior the administration of the radio-labeled compound. And we've presented during the SNMMI earlier this year the result of a sub-study that was designed to determine the impact of that cold antibody injection on PK and safety profile. And we have seen a favorable gain in tumor lesion uptake against the normal tissue using that approach of first administration of a cold antibody. Our second program, FPI 1966, for which we've actually announced the successful dosing of the first patient last week, is targeting fibroblast growth factor receptor 3. And FGFR3 is a great target. It's an interesting one because it's a validated target. The FDA approved already a pan-FGFR3 inhibitor. And also there are strong evidence of target expression for potential indications that are extremely appealing, such as bladder and ovarian cancer. The latest addition in our clinical portfolio is a good example actually of the superiority of Actinium-225 against beta emitters. And the product with recently been approved for, that received the IND clear by the authorities, is FPI 2059. It's actually a small molecule that targets NTSR1 receptor. And originally that was a compound that was already present in the clinic and that was studied as lutetium-177, so beta emitter version, which was really showing very good image. And we were seeing lots of positive tumor uptake on NTSR1 expressing tumors. And Fusion did the conversion of that compound into an actinium-based compound. So we have regional formulation, extended the shelf life, and we worked on the characterization of the compound in order to get the IND clear with the FDA in June of this year. And through our preclinical model, we've demonstrated that we actually achieve a single dose tumor kill, and we have a tumor uptake that can, a superior tumor uptake or equivalent tumor uptake to the lutetium compound using 1500 for a lower dose with our actinium equivalent. As I said previously, Fusion is also working on bringing up or trying to better position a targeted alpha therapy in the conventional toolkit of the oncologist. And in order to do so, we are pursuing combination trial with conventional therapies. So we are combining actually our radiolabel compound with immuno-oncology agent. And in preclinical models, we have observed abscopal effects on distal lesions, which makes that combination of a great potential. We've also worked with DNA damage response inhibitor that we have combined with our first Actinium-225 radiolabel compound, FPI 1434, and there we have observed a great synergistic effect between the two molecules, which paved the way essentially for a potential dose sparing of both agents. As mentioned, we are collaborating with a larger pharma organization, and one of the programs we've announced this year is coming from our collaboration with AstraZeneca, with which we have a partnership to actually develop not only novel targeted alpha therapies, but also studied combination trial with some of their IO agent and DDRIs. So we have released our first target earlier this year for a bispecific antibody that Fusion is developing and radiolabeling with Actinium-225. And this program is right now into IND enabling phase. That's it for me. Thanks everyone for participating.
Thank you, Kevin. We'll go on to our next presenter, Ken Song from Razi Bio.
I'd like to thank the DOE for the invitation to sort of share our experience working with Actinium. And just briefly, my background is I'm a physician originally by training. I left medicine 15 years ago and have been on the investor and entrepreneurial side for the last 15 or so years, and helped get RayzeBio started just about 2 years ago. And must say that this is by far probably the most interesting and fascinating thing that I've been involved in my entire career. So a little bit just about who we are. You know, we are a private company based in San Diego. Our focus is to work on novel and innovative targeted radiopharmaceuticals with an exclusive focus on solid tumors. You know, being a private company, there's not that much information out there about us, but happy to share with you a little bit more about sort of what we've been up to over the past couple of years. From a pipeline and capabilities perspective, we have built what we regard as a sort of first-in-class pipeline that really spans all the way from the earliest parts of discovery, now all the way into sort of clinical stage programs, with one program potentially entering registrational studies with Actinium next year. And that drug is RayzeBio, which is Actinium-225 that is being delivered via a binder called dotatate. And dotatate, some of you may be familiar with, because there are both approved imaging agents as well as therapeutic agents that utilize dotatate, albeit with different radioisotopes. RayzeBio is currently enrolling in a phase 1b study. That's actually a combined protocol as a phase 1b/3 in patients with gastroenteropancreatic neuroendocrine tumors. And we're also evaluating other clinical indications as well for RayzeBio. Beyond that, the genesis of the company was really focused on novel discovery for this field. And we now have 3 novel small binders. These could be small molecules and peptides, which is our focus, that are all focused on delivering Actinium-225. And those should begin clinical imaging studies in the very near future. A lot of the work is coming out of our research and development facility that we built here in San Diego. It's a bespoke facility that has all the capabilities to take care of radiopharmaceuticals discovery. Paired with that is actually a commercial scale manufacturing facility that we're building in the Midwest that should be completed in the second half of next year. Despite starting off as a startup company just 2 years ago with just 2 people, we've now expanded to nearly 80 employees, and we're continuing to grow. A lot of us come from a background of having been involved in other companies and bringing other multiple products into the field, although many of us are newer to the radiopharmaceutical sector. And so we've complemented that, though, with several key individuals who have had a long history and career within radiopharmaceuticals. We've raised a fair amount of money since our inception back in August of 2020. And there should be another announcement forthcoming shortly here, highlighting additional capital that we've been able to raise, which I think just further sort of highlights the interest and excitement that's coming more broadly into the radiopharmaceutical sector, particularly around Actinium-225. This is a snapshot of the pipeline. This gives a very high-level overview. I'll spend a little bit of time talking about RayzeBio, which is actinium dotatate, and our work in neuroendocrine tumors. And you can see beyond that, there is a slew of sort of disguised programs using either novel peptides or small molecules, which we've discovered through our internal efforts, that are going after a multitude of different cancer types. So the lead clinical indication that we're pursuing are neuroendocrine tumors, particularly of the GI tract and the pancreas. Some of you may be familiar with this without really knowing it, because actually Steve Jobs, the founder of Apple, had actually died of a pancreatic neuroendocrine tumor. The vast majority of neuroendocrine tumors overexpress the somatostatin receptor 2, and one can actually therefore target this receptor to provide targeted delivery of different radioisotopes. Now, prior to the approval of a drug called Lutathera, there actually had been very limited options for treatment of both GI and pancreatic NETs. And the expectation here is that all these patients will eventually progress. There is a radiopharmaceutical named Lutathera, which takes advantage of dotatate, which is a somatostatin analog that binds to the somatostatin receptor, as well as lutetium, which is a beta particle radioisotope. And the clinical data with lutetium dotatate is actually quite impressive. Here is plotted a progression-free survival curve, and it was based upon this data that lutetium dotatate, known as Lutathera, was actually approved both in the US as well as abroad in Europe. Patients that were treated with Lutathera are represented on the blue line, whereas those that received control therapy with long-acting somatostatin analogs without a radioisotope are shown in the red. I think one can easily appreciate the dramatic survival benefit that is seen here with lutetium dotatate. Now, despite this impressive separation, unfortunately, as you look towards the right-hand side of this graph, you will see that patients do continue to progress. And actually, as time goes on, virtually all patients will progress despite being treated with lutetium, the beta isotope dotatate therapy. This drug is on the market. It's been approved for the past several years and is currently marketed by Novartis. Now, an interesting thing about neuroendocrine tumors is that it's a relatively indolent and slow-growing tumor. And so despite patients progressing after having received therapy with lutetium dotatate, their overall survival is actually much longer, and there's a gap between the progression and the survival. And that's depicted here on this graph. Here, what we're showing you in the red is the median progression of patients that have pancreatic or GI neuroendocrine tumors. And you can see that in general, there's progression of about 29 to 31 months on average after treatment with lutetium dotatate. However, despite the progression, you can see that most of these patients continue to survive for up to several more years. And therefore, there is this window, an opportunity to bring forth some other therapies to treat these patients. However, there is nothing available today to actually treat these patients who have progressed after treatment with lutetium dotatate. So that's why we sort of see this opportunity to fill this gap by developing RayzeBio, which is actinium dotatate. And as you can appreciate here on the left-hand side of the slide, you will see that Lutathera and RayzeBio are similar in the fact that they utilize the exact same binder dotatate. The difference here being the isotope, where RayzeBio takes advantage of Actinium-225, whereas Lutathera uses the beta isotope lutetium-177. Our strategy from a development perspective is to evaluate RayzeBio in neuroendocrine tumors in patients that have previously been treated with Lutathera and that have now progressed. We're utilizing a similar dosing frequency, and obviously we're able to leverage clinical centers that already have experience with radiopharmaceutical therapy, given that we're going after patients who've already been treated with Lutathera. We will pursue orphan drug designation, and we've also been able to secure some IP, intellectual property, on formulations around RayzeBio. Here I just wanted to sort of show you that the formulation development for actinium dotatate was actually fairly complex. Actinium and lutetium have distinct properties, and given that, the radio labeling and the stability conditions are not easily transferred from lutetium to actinium. You cannot use the same conditions that you would use for lutetium labeling. We actually had iterated on this numerous times, I think over 100 times, to explore a variety of different formulations, and ultimately came up with a unique formulation that allows RayzeBio to have very strong radio labeling efficiency and stability. Below here on the bottom of the slide shows you a graph in terms of the early formulations that we had, where you can see this TLC tracing, where we had, at about 5 days or 120 hours, you can see in the early formulations, you see that about 36% of the drug had broken down, whereas our current optimized formulation, even out to 5 days, we're able to see about 98% of stability of the labeled drug product. This then becomes amenable to a centralized manufacturing strategy for broader distribution. Now, an interesting feature of this drug is that there's actually clinical data that's already out there that demonstrates efficacy of actinium dotatate, including in patients who have previously been treated with Lutathera. On the left-hand side of the slide, you will see a comparison between objective response between patients who have been treated with lutetium dotatate, as depicted in the gray, as compared to those patients who have received therapy with actinium dotatate, which is depicted in the turquoise bars. And you can see that whether it be radiotherapy naive patients or patients who had been retreated after Lutathera progression, you can see that the actinium dotatate clearly outperforms in terms of a response rate. And on the right-hand side of the slide, you can see that actinium dotatate also is able to see a much better progression-free survival as compared to retreating with Lutathera in patients who had already received Lutathera treatment and then had progressed. Now, the reason why clinical data already exists is because within the radiopharmaceutical sector, there is the potential for clinicians, particularly outside the US, to utilize research compounds as compassionate use. This is actually how many of the drugs that are on the market today, including the PSMA as well as Lutathera, were first evaluated and developed. Similarly, there has been some clinical experience with actinium dotatate in different academic medical centers outside the US. And obviously this gives us high confidence that we can take this forward and have a approved therapy that could be more broadly disseminated. The other thing that was nice to sort of verify is that actinium dotatate is quite well tolerated. We were able to look at a series of laboratory data in which patients who had already received a full cycle of Lutathera and then went on to receive treatment with actinium dotatate. We're pleased to see that there were no significant toxicities, at least based upon clinical laboratory criteria. And here on this graph, we're just showing you creatinine as a surrogate for renal function and platelets as a surrogate of bone marrow function. to show you that regardless of the total amount of actinium that was administered, you could see that there's been really no deviation between the baseline values as well as the last values that were obtained based upon the data cutoff dates. So we are developing RayzeBio in something called the Action-1 study. This stands for Actinium for Neuroendocrine Tumors. This is a streamlined phase 1b to phase 3 trial. The phase 1b trial is currently underway. This is a dose de-escalation study design. We have already dosed a fair number of patients at the top dose of 120 kilobecquerels per kilogram of actinium dotatate. We are monitoring these patients for any dose-limiting toxicities for an 8-week period after administration of RayzeBio. Following the completion of this phase 1b study, we have already designed a pivotal trial where we will do a randomized evaluation of RayzeBio against a standard of care menu options. And here the primary endpoint will be progression-free survival. This will be a global study, and you can see a reference to the clinicaltrials.gov identifier on this slide here. This study is being performed in patients with GI and pancreatic NETs after they have been treated with prior lutetium-based somatostatin analog therapy. Just a couple last slides here. I just want to give you an overview of our sort of discovery workflow that we do to identify novel radiopharmaceutical therapy oncology drugs. First and foremost, it starts at the top of identifying those targets which are selectively overexpressed in tumors, but we have then done a systematic evaluation of a multitude of different binder moieties and then subjected them to a pretty intense screening funnel that is a combination of both in vitro as well as in vivo screening. This is all being done out of our research center here in San Diego. And I would say one of the things that we've come to appreciate is that the in vivo data is absolutely critical to be able to understand what properties of these binders are going to be appropriate to take them forward into clinical development. We've actually conducted over 200 in vivo studies since our inception. I would say this in vivo data has been absolutely critical towards being able to guide our discovery efforts to build out our pipeline. I want to just show you here quickly some preclinical data on a specific cell surface target that we're working on that is overexpressed in a multitude of different solid tumors. We have developed a novel small binder. It's actually a small molecule that has exquisitely high affinity and selectivity against this target. And then we subjected and compared this, in terms of efficacy between actinium and lutetium. You can see here in this specific tumor model, this is a xenograft model, and all these are single dose injections. You'll appreciate on the left-hand side that you see a dramatic benefit when you use the binder and you deliver actinium. And that's depicted by the green line. You can see that nice separation from the vehicle. However, you can see that lutetium being dosed at 1,000-fold higher did not have any impact on the tumor growth, highlighting the advantages of actinium over lutetium. However, using the exact same binder, but now in a different tumor model, you now can see that actinium and lutetium actually behave similarly in terms of their efficacy, both by seeing nearly complete tumor growth inhibition. I think this just further highlights that the preclinical discovery and work being done to identify novel binders for radiopharmaceuticals is fairly complex, and it does take an understanding of both biology and chemistry and those two groups working closely together. So in summary, I think we're all here because we're interested in actinium. Actinium has already demonstrated robust clinical safety and efficacy. I think new radiopharmaceutical drugs that can leverage the potency of actinium can really have a substantive impact on the lives of cancer patients. But we're still in the early days, and I would say the continued collaboration amongst industry, the DOE, and others, we need to work together to be able to advance these therapies forward, not just only in development, but also into full commercialization so that as many patients as possible can have access to these great therapies. Thank you for your time.
Thank you, Ken. We'll move to our last presentation, and then it'll be given by Monideepa Roy from Actinium Pharma.
Thank you, Cathy. I'll share my screen. So thank you for this opportunity. It's great to be here and just learned so much from the prior talks. So here, I'll give you a bit of a background about what Actinium does, and then I'll focus on some of our key programs with Actinium, Actinium-225, that is. So our company is a leading specialty oncology radiotherapeutics company, and we have several compounds in our pipeline. Our lead program is IOMAP-B, which is basically apamistamab targeted against CD45 and conjugated to iodine-131. This is a paradigm-shifting conditioning agent that recently completed phase 3. We are just awaiting the top-line data. But we think that for elderly relapsed refractory AML patients who are otherwise deemed unfit, will have IOMAP-B that'll change their treatment paradigm. So with the data that's eagerly awaited, we hope to go for BLA filing and then approval and launch and commercialization. We already have a commercial partner in EU and MENA geographies, and we look forward to taking this forward further. And then also with apamistamab conjugated to iodine, we have 2 other pipeline programs, mainly in CAR-T and gene therapy, which are also moving along with a lot of promise. What I'm going to focus a lot on today is our second program in the clinic, which has completed phase 1. This is Actimab-A. This is a CD33 targeted antibody, lintuzumab, and we have explored this in combination with the salvage chemo, CLAG-M, or with the targeted therapy, venetoclax. And we have some very exciting data that I would love to share with the group, and how we are planning to take this program forward. And also, we have, evolved our R&D programs in a way that we support partnerships and collaborations with our platform technology with companies such as Astellas on theranostics programs on undisclosed targets so far. We have a very exciting HER3 antibody radioconjugate program in partnership with a company called Aveo, based out of Boston. And also we explore immunotherapies in combinations, and we have a partnership with a company called Epicentrix for their small molecule inhibitor against CD47. We are basically targeting agent agnostic. We look at antibodies, although our pipeline is largely focused on antibodies. We also look at other modalities. And we are radioisotope agnostic as well. The 2 of our programs in the clinic, the lead program is a beta emitter, iodine-131, and then the follow-on program is an alpha emitter, Actinium-225. We have several discovery programs with lutetium-177 as well. So as I mentioned, I'm going to focus our discussion today mostly on Actimab-A, which is CD33 conjugated to Actinium-225. We recently presented some very encouraging data in ASH last year. We have, several clinical trials running, more than 150 patients that have been tested with different combinations. And we see a dose-dependent response. We feel that, there are a lot of advantages with the differentiated mechanism of action of our CD33 targeting antibody radioconjugate that can really benefit AML patients. So some of the results that we showed last year at ASH, shows with the combination with the salvage chemotherapy CLAG-M, we see 72% MRD negativity rate in relapsed refractory AML compared to only 39% with CLAG-M alone. We saw a dose response across all cohorts showing 80% response rates. And we really think that, this can shift the paradigm for treatment with salvage chemotherapy. We have some very encouraging data also with venetoclax. This is, as you all know, is a BCL2 inhibitor, and particularly in patients that carry P53 mutations, we saw 60% ORR. We are now advancing to the higher dose of Actimab-A, dose cohorts, and we hope to get, continued data this year itself. So the high MRD negativity rates and also the ORR in patients with prior venetoclax therapy support the continued development of our Actimab-A programs. So as you can see that Actimab-A can be used for fit relapsed refractory AML patients in combination with CLAG-M, as also unfit relapsed refractory AML patients with venetoclax and other such targeted therapies. With these findings, we believe that Actimab can have a lot of synergistic effects with other therapies. So the current active programs are, of course, with chemotherapy and with targeted agents such as venetoclax, but we're also exploring Actimab in cell therapies and in combination with immunotherapies. So these are some of our trials that are currently running, with CLAG-M, venetoclax, azacitidine, with azacitidine, and also with the CD47, we are planning to generate, some data and file the IND and move that forward. This particular program, we have really received the support of DOE for maintaining the supply of actinium. So there are also some other programs where we have explored actinium. So our lead program where we have targeted CD45 with abemaciclib, there we have also explored conjugation with actinium in our preclinical studies of multiple myeloma. And here in this set of data, you can see that the single dose treatment of these animal models carrying multiple myeloma tumors with actinium conjugated CD45 antibody completely ablates the tumor growth. Compared to control, the effect was really significant. We also tested other targeting agents in multiple myeloma. We looked at daratumumab conjugated to actinium in this particular Dowdy xenograft model. And here also we saw significant tumor ablation with the actinium conjugated dara. With this, with all of these results, we thought that some of the targeting agents could also benefit solid tumors, as showcased by, the talks just prior to this one. And we looked at a HER3 expression across solid tumors, and we thought that, this is a great target to go for. And then we generated a HER3 antibody radioconjugate, which is first in class. And you can see that in a model of non-small cell lung cancer, this particular HER3 ARC completely regressed the tumors. And when we tested this particular model in combination with CD47 inhibitor magrolimab, in this particular graph on the very right, you see magrolimab alone shown by the dotted red line that doesn't really have an effect in the NSCLC model that we tested here. But the solid red line, which is a combination of magrolimab with HER3-ARC, shows considerable tumor shrinkage and shows a lot of promise with this approach. So even in our solid tumors, we are looking at actinium conjugated radioconjugates that can have a lot of options as a single agent, as well as in combination with immunotherapy. And then, as I mentioned, we have some undisclosed targets with Astellas, which we are also moving forward with in our discovery and preclinical programs. So, we feel that, we have made a significant amount of progress with Actinium-225 radiotherapies. We proudly hold a leadership position here with more than 100 patients dosed with Actimab-A. This is our clinical stage Actinium-225 conjugated ARC. And then we are poised to move Actimab-A into the next phase as a backbone therapy for AML patients. This is really a high unmet need, and we hope that it can really create a difference. And to bring all of these efforts into fruition, we would require, a high quality and continuous supply of Actinium-225. We cannot thank DOE enough for the ongoing support that we have had so far. But we are looking forward to working with the entire community as well as DOE to figure out how we can take this commitment further and make this supply one of our one of the challenges that we can overcome together. So with that, I would like to end my talk here and thank you once again for this opportunity.
Thank you. Thank you, Monideepa. If all of the speakers will, I guess, come back and turn your cameras on. So I just want to thank you. It was very interesting. And it was really great to see all the progress that you've all made. So it's pretty exciting. So we have a number of questions here. I do think there are some questions about the slides. I believe the slides will be posted on the NIDC website. Some of you asked some business questions about cost and pricing, and you really need to take that to the NIDC. They're the business people, and I'm not. And I get in trouble if I talk anything about business, so I stick to science. So then we'll try to go through some of these other questions. The first one asked if there are ongoing discussions with NRC and FDA. Are there any plans for similar discussions, workshops with ex-US regulators? I think they're saying regulators to gain similar acceptance of thorium-generated Actinium-225 to support clinical trials run outside of the US. So I'm not currently aware of anything that the Department of Energy is doing. I do know that the IAEA has somewhat interfaced and brought people in to talk about this, but I'm not aware of any other ongoing efforts. A question is can you contrast and compare bismuth-213 and Actinium-225? Actinium-225 is about a 9.92 day half-life. It decays, giving off 4 alpha emissions. The bismuth-213 has about a 47-minute half-life, and it is actually derived from an Actinium-225 generator, and it has a 47-minute half-life. So due to their half-lives and nuclear properties, they're often chosen for treating different types of diseases. Based on your experience, what is the limit of Actinium-227 and the drug product that the FDA or EMA will accept? I have no experience that I can speak to that. I don't know if any of the other presenters would like to speak to that.
We can't. I mean, for us, we've only been working with Actinium-225 that does not contain the 227. So I'd actually be curious if anyone else has an answer to that on the panel as well.
I may not provide a complete answer to these questions, but it will really depend on which type of compound you are talking about, the formulation, the injected dose. Establishing a limit for an impurity will vary depending which product you are going to be filing with the authorities. So I'm not sure there is it's a pretty much open-ended question here. We could debate for a long time.
I guess, Kevin, I'm just curious, but have you had success in using the accelerator produced actinium in your final drug product and getting FDA comfortable with injecting that into patients?
We have a path with the FDA to look at the use of accelerator-based material. It's not something that we are doing right now in the clinic, but we are into IND enabling studies in order to do so. So we've met some of our milestones in terms of CMC and formulation development.
Monideepa, do you have any experience with that?
No, so I was just going to mirror you, Ken, that all of our experience has been with Actinium-225 so far. So we don't have any information about 227 and love to know more from the community.
Okay, thank you. Thank you, Cathy, for the presentation. I would like to know, are you allowed to use 50 grams of radium-226 as a target for Actinium-225 production? So I'm not sure that I'm allowed to answer any details about how we're bringing up that process. And I guess what I would like to point out is that, the alternatives that we're looking at, we're really looking at the feasibility of bringing these up. And so we're not really at a point that we're providing this material, or that we've shown that they're viable methods that are actually going to be supportive. So I would just dawn you to keep that in mind. We have a lot of work to do on these to really get them to the point that they could even be looked at to be thinking about supporting clinical developments. And I was asked about recycling and containing. I can't really speak to that either. What is the acceptable regulatory threshold ratio for Actinium-227 and Actinium-225? What technique is used to assess the ratio? So I think part of this question is similar to what we attempted to answer the first go around, so I will leave you to that. As for the ratio, we have looked at this, and we more or less have been using ICP and gamma ray to really look at these ratios, and that's how we derive it. I assume this question is for someone else. For TME studies, what kind of actinium was used, generator or accelerator produced? Gary, this may be.
Yeah, that may be for me. So we use the accelerator produced. Later this year, actually, we should have some generator produced, and we're going to be comparing in vitro and in vivo the prior studies to the new studies. So hopefully we have something to share on that later.
So Gary, I think this question is for you as well. Have you performed secondary assessments of neutral cell health and mouse cognitive function during the radiopharmaceutical therapy?
Not really. We do the CBC, and then we do check mice, routinely almost every day for, cognitive function, but they have a brain tumor. And so we couldn't even distinguish at this point what would be from the radiopharmaceutical therapy and the brain tumor. So we don't have a good answer.
Thank you. And I think the next question is for you as well. What are the thoughts as to why pre-dosing cold antibody leads to increased uptake and tumor?
Okay, so I'm just speculating. Initially, for myeloid cells that we're targeting, these immune cells, there's a lot of them circulating. And so we may be just blocking some of the receptors on circulating cells. The other thing is we might be, if these cells are migrating and carrying antibody with them, we may be blocking some of the receptors on each cell and spreading the tracer for more cells that ultimately migrate to the brain.
Okay, thank you. So then the next question says, what are the advantages of using cerium-134, lanthanum-134 versus yttrium-86 or gallium-68 as PET imaging partners of Actinium-225? I think there are probably cases where you can use yttrium and gallium, but they have very short half-life. So if you're trying to follow an antibody, that short half-life is really going to be a problem, and it's not going to really work. So you need something with a longer half-life that's going to allow you to really image when the target actually, or when the antibody actually reaches its target. I would also say that the cerium and lanthanum, it seems to be that their chemistry is more mimicking what we've seen with actinium. So it seems like the distribution seems to be better, and some of that's actually been demonstrated in the literature. So that's really what the advantage is of using that. So the next question, thanks, Kevin. May I know which approach or reaction is Fusion using to Actinium-225 production?
Thanks for the question. So I think it was released last year with some partnership right now to study the or to look at thorium spallation route and the P2N reaction. We're using Iridium-226.
Okay, so for the next question, it says, are there any SBIR, STTR, or other DOE extramural grant mechanisms available to assist new startups and current efforts to make commercially relevant quantities of Actinium-225 widely available? So the Isotope Program actually does not participate in the SBIR, STTR program. However, in the upcoming Accelerate initiative, for which they have modest funding in 2023, industry and universities may partner with national laboratories and translational research to advance the readiness of isotopes produced by the DOE IP, to advance the isotopes to readiness in clinical trials, dosimetry, toxicity, or labeling. And I would assume if you want some more information on that, you could probably also reach out to the NIDC. The next question is, it was mentioned the Actinium-225 FPI drug candidates shelf life was improved. What is a typical shelf life for Actinium-225 drug?
And I think can partly answer that question when it was asked. Obviously, if you want to consider a central production scheme, you need to look at shelf lives that are 5 days and plus in order to be able to centralize your manufacturing operation and be able to distribute the product across your different clinical sites.
Thank
I mean,
you.
just as a reference, you know, I think Lutathera's label has a 72-hour shelf life that is centrally manufactured, and that's been successful. I do think if you can extend that, obviously that just makes it easier from just the logistics standpoint.
Mm-hmm.
Thank you. So the next question, how do you reconciliate the issues of translating preclinical data to first in humans, especially due to lack of proper mice models?
I mean, this is not unique to radiopharmaceuticals. I mean, this is a problem for any modality across any disease state where you're always trying to correlate findings in the mouse and ultimately go into humans. And I think this is why you see so few drugs actually make it through clinical development, is because that translation is usually poor. But I would say one of the advantages with radiopharmaceuticals is that you at least can conduct some type of dosimetry analysis in these animal models. And if we at least look at some of the examples for drugs that have been approved, like with PSMA 617 and with dotatate, there has been a reasonable correlation between mouse and humans. Whether or not that will be replicated with these other newer investigational agents is still to be determined. But at least there's some precedence and also the ability to use dosimetry, which you cannot do with other small molecule or standard other type drugs, to know whether or not you might actually be able to see efficacy and safety once you go into human testing.
I think another route is to use companion animals. They often have similar diseases that humans have. I know when I was at the University of Missouri, we used to do clinical trials in companion animals on some disease states, and it seemed to kind of fairly closely mimic what you would see in humans. So that might be an alternative. I should point out, I had directed people to the NIDC for that grant opportunity. I've been corrected, and you should really reach out to Ethan Balkan if you're interested in grant funding. The next question, you've all highlighted the benefits of RLT and solid tumor targets. Can you discuss the potential benefits or complications of RLT in liquid cancers?
Some of the RLTs are to do with, as expected, with myelosuppression. So therefore, cytopenia. But those are basically, very manageable within the clinic. So with Actimab A, we haven't seen toxicities where anything was beyond grade 3 for myelosuppression. And even whatever, we have seen was managed in the clinic in a reasonable way. So there are some obvious toxicities that will emerge, but with the current support for the patients, and particularly for those patients who have run out of options, such as, unfit, who are deemed unfit for intensive therapeutic treatment and have no other option, this can be really life-changing, paradigm shifting for them. So, it's always a fine balance, toxicity and efficacy, but depending upon the particular patient who is coming in, and then, as pointed out, we do individualized dosimetry to figure out exposure to other organs, and then based on that, we see great benefit to patients with RLT.
Thank you. Any other comments? Okay, so the next question. Can you elaborate on the statement in the first presentation, related challenge related to Actinium-225 concerning perception and regulatory compliance remain, and the plans to address these challenges? So I think that we continue to try to let people know about the studies that we've done to look at the 227 for the dosimetry, and we try to continue to tell you where we at with the production. We also are working to try to address some of these challenges. I mean, as I mentioned, gallium-68 generators, one of the challenges was really the germanium-68, and the NRC ended up giving an exemption. And that's, there are other isotopes that have been used that have long-lived impurities that have been dealt with the NRC. And so we are attempting to address these as we go forward. So correction, based on your experience, what is the limit of Actinium-227 in the accelerator-derived Actinium-225 drug product that FDA EMA will accept? I think we kind of answered this as well as we can before. So I don't think we can add anything more to that. To all the developers of new RPT approaches, are you considering individualized treatment planning based on radiation dosimetry?
I think for the field in general, we need to walk before you run. This idea of individualized dosimetry sounds quite appealing because you can marry the diagnostic with the therapeutic, but it's impractical when you think about just the infrastructure and the resources that are available there, and even for the patient experience to go through a specific dosimetry plan before their treatment. I think many of us are doing dosimetry analysis to understand the broader dosimetry profile of the molecules. But I think the idea to do true individual patient-specific precision medicine, I think is still a ways off. I mean, I think it's interesting academically, but I don't think it's practical for full-scale development and commercialization.
Other thoughts?
Yeah, I agree with what Ken said, and although as a physicist who spent his time in radiation oncology before, where patient treatment planning is key and is absolutely necessary, in this field, there might be a better use of it to find patients where you can really use dosimetry to find an added benefit and identify patients who don't have as much benefit of your therapy, and really tailor, maybe not individually at the exact patient level, but tailor prescription levels of treatment and really find optimal strategies that still are practical and logistically manageable. But it's a very interesting debate in the field that is as 50-50 split as anything can be right now. but it's interesting.
Okay, thank you. So is there any plans to increase radium-226 supply for actinium-225 production? I believe that the DOE is now supplying some amount of radium-226, but my suggestion would be is that you reach out to the NIDC website and ask them about the supply. And I would say for the next one, which is asking how much can be available for low energy cyclotron irradiation work, that the radium-226 questions, you should really reach out to the NIDC. I don't have any real knowledge on the availability. Would any of the companies be willing to share their desired specifications for actinium-225 shipped to them as an API?
I mean, I think a lot of us, this is why we're invited. We're using the material that's been provided by the DOE. And so I would say, for anyone out there that is interested in producing actinium-225, if you meet the DOE specifications, obviously we've been able to carry that forward into clinical development.
Okay, thank you. So there's questions regarding the specifications of the generator versus the cyclotron material. If you actually go to the NIDC website, they have specifications on there and you can download those and look at it. So I would recommend you go there. Cathy, thanks for noticing that Modulation Therapeutics has closed metastatic uveal melanoma patients recently. Okay, you're welcome. Any news regarding the Actinium-225 user for prostate cancer like xerostomia, maybe new methods or any kind of preventative procedures?
I mean, we're not working on this, but I know just by following the industry, I know that people are looking at, there's antibody-based agents against PSMA. There's obviously other targets one can go after. I think this question is stemming from the fact that PSMA 617 and other analogs to PSMA 617 seem to have an unexpected higher uptake in the salivary glands, and experience with actinium leads to debilitating xerostomia. Obviously, if you have a different binder format like an antibody out there, that has been shown at least through imaging and early clinical data to avoid some of that. People are trying to think about different dosing regimens, or but also their targets too to go after.
Yeah, I think last year at this meeting, Neil Bander talked about some of his J591 work with the antibody that they don't see xerostoma. I do know people have been working with albumin binders to try to address this as well, but I'm not an expert on this. Question for Cathy. You mentioned in your opening that actinium-227 quantities and the DOE accelerator produced material are inconsequential from a dose safety perspective. Do you have any sources or citation for that claim? This seems to be pretty strongly contradict most of the literature I've seen published evaluating dosimetry concerns. So I base it on some of the papers that came out of the work that was funded. I think you can go to the website and those papers are there. Some of these papers came from Kate Dadachova. Also, there was some work that was done by Jeff Nornberg. So that's what I'm basing my answers on. What is the biological half-life of actinium-225 from accelerator produced source? Well, I'm not sure that I completely understand the question, but I don't think the biological half-life is different from the accelerator versus the generator material. And there are studies out there where they've looked at what happens when you actually just inject the free actinium. So I would suggest that you go look up that data. Question to Cathy, do you think that the supply of radium-226 as starting material could be as difficult to source as the ytterbium-176 is? I'm not sure that I'm in a position to really answer which one of those types of material will be more difficult. I would assume that since radium-226 is not is mostly being harvested right now and not being produced, that it is difficult. But my understanding is that there are ways that they could be producing it if the demand gets to that point. But I'm not really an expert on radium-226. Do any of you radiolabel actinium at room temperature? Is that a no?
Yeah, we
I'll
do.
turn over to people that work with antibodies. I mean, for peptides and small molecules, there's a lot more flexibility, and so we haven't needed to do that. I'm not sure about Fusion and Actinium Pharma.
So
we primarily work on lots of antibodies, so we don't have that luxury at the moment.
okay. Answer to previous questions. Radtron LLC is under NIH SBIR for production of radium-226 to support actinium-225 production. So there we go. Do antibodies typically cross the blood-brain barrier? I don't think they did. How do you suggest that your compound does this, especially with a highly charged middle inside of a chelator?
Yeah, that's for me. So, I mean, that's a great question. And we, there's two things. Number one, for in GBM, the blood-brain barrier is damaged. But more so, our approach is specifically targeting the myeloid cells that are continuously fed from the blood into the tumor. And so we think it's possible, we have no data to support this, but that the myeloid cells take up the antibody and then continuously are trafficking into the tumor to replenish the other ones that are dying. Just the life cycle of macrophages, monocytes.
Okay, the next thing is a comment, so I'm going to avoid that. What ways are cerium-134 currently produced? Any other radioisotopes that can act as match pairs for actinium-225? Los Alamos has brought up a method that they're evaluating, irradiating lanthanum targets and then processing that to actually isolate the cerium-134. And I guess I should further point out that we're trying to transfer that method to Brookhaven so that in the future it could be made available from either site year round. What other isotopes can act as match pairs? And I think that somewhat depends on what you're doing and what the half-life is that you need. Cathy, can you please comment on the amount of actinium-225 produced when using a radium target versus a thorium target? So everything I've seen tends to indicate that there is no actinium-227 that's produced when you use a radium target. And I posted in the slides the amount from the thorium target. And I think that agrees actually with what's in the literature. And that was our last question. Monideepa, did you have something you wanted to say? Okay. Since we have no more questions, I don't know if the panel would like to say anything before we close the user meeting.
I just think it's great that we have these meetings and that we're able to share things. I'm not sure if the plan is, I guess the plan is to continue these virtually. Obviously, some of us see each other at conferences and the like, but I do think, again, working together and I think working closely with the DOE, again, very appreciative for all the different efforts I think that the DOE is doing in terms of its different exploration on methods of production. I know that we're all rooting for you because I think actinium continues to be in constrained supply. And I think the accelerator produced material is interesting, but I think if one is able to have larger quantities of actinium without the actinium-227, that probably just makes it easier from a development, a waste handling, and other perspective.
Thank you. Any other comments?
No, just would like to thank you for your continued support of our programs. And, it's so great to have the community come together like this, and look forward to other opportunities where we can brainstorm and discuss.
Okay. Okay, well, if there's nothing else, I want to thank you all. It was really nice to see how you're progressing and all the work. I know it's very challenging, but I know we're rooting for you as well. So it looks like maybe we got, okay, no, we just got to thank you. So, all right, so on that note, I guess we will close the user group meeting, and I just want to thank everyone for their participation.